Hello and welcome to Proteome Informatics. Today we're going to cover 6 different topics, all strung together in one long video, but we've broken it up into about 20-minute segments that we're going to walk through bit by bit to increase our understanding of how we identify peptides and thus proteins in proteomics and how we quantify them. I'm your host, Dave Tabb. Nice to meet you. I am currently a visiting scientist at the Mass Spectrometry for Biology unit at Pasteur Institute here in Paris, France, but I'm actually, as you can probably tell from my accent, from the United States. I worked at Vanderbilt University for a long time there, and for the last 5 years I lived down here in South Africa. So I've been a bit of everywhere, and I have really enjoyed the subject of proteome informatics, and I thought this would be a good opportunity to share it with you. So our first segment, should span around 20 minutes, is relating a peptide sequence to its tandem mass spectrum. This is a topic that I feel is really important for everyone to understand, and yet it's something that we frequently skip by. If I have a particular peptide, why is a tandem mass spectrum informative to me in discerning what its sequence is? So let's walk through that. First off, most proteomics laboratories today make use of a bottom-up or shotgun technique for producing tandem mass spectra to represent the peptides in a complex mixture. But typically, biologists are not starting with a mixture of peptides, they're starting with proteins. So if you have 10,000 proteins in a sample, you probably will end up digesting it to produce an even more complex mixture of peptides. The reason for this is that peptides are a lot easier to separate, they're a lot easier to fragment, and they're a lot easier to identify in the end. So having made a protein mixture more complex by digesting it, we may then separate that very highly complex mixture, 100,000 peptides or more, into different fractions. This can be carried out a number of different ways. This lecture is not about that. However, an individual fraction can then be separated over time by the hydrophobicity at which peptides elute from reverse phase liquid chromatography. So reverse phase liquid chromatography is a very widely used method for doing this separation upstream of the mass spectrometer. Now, the mass spectrometer cannot measure things that are not charged. It needs ions to start with. So these peptides are turned into ions by passing them through electrospray ionization, sometimes 3,000 volts or more. So this produces a cloud of charged peptide ions that can then be measured 2 different ways by the mass spectrometer. So all the things in the bottom row here take place in the mass spec. We see that we have a high resolution measurement, very typically, of the peptide ions in intact form. This is something that we call the mass spectrum, typically a highly resolved, highly mass accurate mass spectrum. And from there, we're going to go ahead and do something called tandem mass spectrometry to each peptide. So for this, we're going to isolate a bunch of ions, typically of a single peptide, just many, many copies of it. We're going to cause those ions to collide with gas to cause them to break, and that creates fragment ions or daughter ions, depending on which terminology you're using. And then we can catalog the set of fragment ions that are produced for that given peptide in a single tandem mass spectrum. So for a very complex peptide mixture, over the space of about an hour, we may produce as many as 10,000 or more tandem mass spectra representing the fragments of individual peptides. But there are quite a few disruption steps along the way, and I find that students sometimes struggle with figuring out which one is which. So let's work through them in order. We start with the problem of denaturation and reduction. So we need to denature the proteins. They have these very complex 3D structures. We want them to stretch out. And along the way, we're probably going to need to break the safety pins that hold the protein structure together to break the disulfide bonds to produce free cysteines. So this denaturation and reduction step causes the protein to stretch out, which gives it better accessibility to digestion. And that enzymatic cleavage is the next step. Having started with a long sequence of amino acids, the protein still intact but unfolded, we now want to digest so that particular peptide bonds are cleaved. Now typically we do this through an enzyme called trypsin, but it's not the only option. Sometimes you'll see lyc used that as a kind of a support to trypsin, but you can also use far less specific enzymes for doing the cutting. And finally, there's fragmentation. of these 3 bullet points on screen, the only one that takes place inside the mass spectrometer is fragmentation. Most typically, we're trying to break one type of backbone bond, meaning bonds that fall along the nitrogen-carbon-carbon, nitrogen-carbon-carbon, nitrogen-carbon-carbon backbone. So break one type of backbone bond through collisions, whether if you're using collision-induced dissociation or higher energy collision-induced dissociation, or through making the peptide a radical. You see this frequently used in electron capture dissociation and electron transfer dissociation. Sorry, I skipped ahead. All right, so for this, we are going to produce 2 different kinds of mass spectra, and it's really important that people understand the difference. So at the top, I have an example of a mass spectrum, which is to say that the peaks that we see here represent intact peptides that we could potentially blow apart to create a tandem mass spectrum. So the mass spectrum is showing us which peptides are eluting at this moment in time from the liquid chromatography into the electrospray ionization source. A tandem mass spectrum does not typically represent multiple peptides. Instead, an individual tandem mass spectrum usually represents the fragments of just the ions of a particular mass to charge value in the mass spectrum. So we see that we have a spike up here at 645.84 in the mass spectrum. And these slides, by the way, are available for you to download if you read the description below the YouTube video. So we could fragment this 645 ion by isolating a bunch of copies of just that ion, causing them to bounce against gas molecules, and then measure the fragment ions that are produced from it. So a tandem mass spectrum can be thought of as the song of a dying peptide. We're blowing this peptide apart. These are the fragments that are produced when we do that. So the ions that you see in a tandem mass spectrum do not represent intact peptides, but rather the fragments that result when we blow one apart. Now, there's a bit of an erroneous assumption on the part of most people that if you have a peptide and you conduct CID on it, that you're blowing it apart. And that's a very loose way to talk about it. From a chemical point of view, we have a fairly complex bit of interaction taking place. First off, I would note that since most of the peptide ions most typically the peptide ions we are working with are positively charged, in vacuo, that means that they're carrying extra protons, most typically. So you could imagine that a proton is sitting down here at the C-terminus on the side chain of that last amino acid. Because we use trypsin, arginine or lysine is a more likely place for a proton to sit than other amino acids. But you might instead have it sitting up here at the N-terminus. But once you start adding energy to these peptides, those protons can start moving to less likely locations. Now I want you to pay special attention to this proton I've drawn in red, right above this second carbonyl. See this tiny little red mark? So that's the proton. That proton is causing the electrons in that double bond to sort of cheat towards the oxygen. Because you've got a positive charge, these are negatively charged electrons, they're preferentially up that way, which means that this carbon in the carbonyl takes on a partial positive charge of its own. When that happens, the pair of electrons that are part of this carbonyl down here can swing across and form a circularization event where this oxygen becomes connected to that carbon. And that's not very stable because carbons do not like being attached to 5 things at once. They simply don't. So what happens is that we end up with the circularized part of the ion departing as what we call an oxazolone, and the rest of the peptide ion departs as a smaller peptide ion, essentially. So in this case, we see that the first 2 amino acids are part of the B2 ion of the oxazolone. We call it a B2 because it's the N-terminal ion, and it contains 2 amino acids. But the last two amino acids are departing as the Y2 ion. It's called the Y ion because it's the C-terminus side of the breakpoint, and it too contains 2 amino acids. So the B2 and Y2 are formed when you get this kind of attack across across the second amino acid. So this is the process of collision-induced dissociation. A very strong, a very important term to know about in connection with this is the mobile proton model that was published by Vicky Wysocki's group through a series of papers. Really helped us to understand how peptides break. And it's useful for you to know that B ions do not have the same structure as Y ions, because Y ions are small peptides essentially. B2 ions have this weird circularized piece at the C-terminal end. So many fragment ions may result from the breakage of a peptide bond. In the world of collision-induced dissociation, we most typically think about the breakage at the peptide bond forming an N-terminal B ion, as I mentioned in the last slide, and a C-terminal Y ion. Now here we have a real sequence. This is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 amino acids long. So this 10 amino acid peptide might break at this position between the 4th and 5th amino acid. That would produce a B4 and Y6 ion. The Y6 contains 6 amino acids, the B4 contains 4. Or it could have broken, say, at the peptide bond between these isoleucines that are next to each other. That would have produced a B3 ion and a Y7 ion. What this means is that as we get closer and closer to the N-terminus in where that breakpoint is located, we get smaller and smaller B ions. But on the opposite end of that, the closer and closer we get to the N-terminus, the larger the Y ions get, because they contain one more amino acid with each place that scoots left. Which means that if you have many, many copies of this peptide, and each of them is breaking independently of the others, it may well be that you get some evidence of where this peptide bond falls, what the mass of the amino acids are on either side, where this peptide bond may break, this peptide bond may break. If you're very lucky, you're going to break them all. You're never that lucky, though. Generally speaking, some parts of the peptide will have better evidence than others. A very, very important concept that you really need in order to understand a lot of proteomics literature and research talks is to understand what a PSM is. This is a peptide spectrum match. In this case, we have a pairing of a particular peptide sequence with the tandem mass spectrum that we believe represents it. So it's important, I think, to imagine these PSMs, these peptide spectrum matches, are in fact hypotheses. It's a hypothesis that the presence of a particular peptide sequence in your mixture is the explanation for why this tandem mass spectrum was observed. Meaning that the peptide sequence can allow you to project the locations of particular fragment ions that should have resulted. You can then look in the tandem mass spectrum to see if those in fact are observed. This correspondence is all part of a peptide spectrum match. So typically an individual tandem mass spectrum, if it is identified successfully, produces a PSM. Out of a huge collection of tandem mass spectra, only some will be matched to peptides and thus become peptide spectrum matches. So that's the first segment. We have several takeaways that come from this. Tandem mass spectra reflect the structures of peptides because each peptide ion can break independently of the others, potentially breaking at a different peptide bond than other copies did. B ions have the lowest mass close to the N-terminus, but Y ions have the lowest masses up at the C-terminus. These latters are both present in the tandem mass spectrum, and the sequences from low mass to high mass are in reverse order. So in effect, you have one ladder of B ions and one ladder of Y ions in reverse order laid on top of each other. That makes it very difficult for us to interpret a tandem mass spectrum by just reading the peaks. It's gotten easier in some ways because we have highly mass accurate fragments now, but that is not, generally speaking, the way that we produce PSMs. In the next part of the segment, we will talk about database search algorithms that allow us to ask, does this peptide sequence match well to this tandem mass spectrum? This is your cue to pause the video and go take a moment away. When you come back, we'll be fresh again. Welcome to segment B. In the last one, we talked about why the fragment ions of in a tandem mass spectrum can be related to a peptide sequence. Now we're going to explain the algorithms that match peptide sequences to tandem mass spectra. This is carried out through the sequence database search algorithm. I would note that a lot of what the computer must do to identify proteins overall is to recapitulate the steps you did on the bench. That you started with a mixture of proteins, you digested it up, and then you told the mass spectrometer to collect a bunch of tandem mass spectra. In reverse, the computer must match those tandem mass spectra back to peptide sequences, which is the database search algorithm. It needs to determine which peptides were confidently identified and which ones were not. That is the target-decoy method that we'll talk about after this. And then finally, it needs to infer a set of proteins to explain the peptides that it saw. That's the segment after that. This diagram is probably the most important one for understanding the key algorithm that makes proteomics work. It is the database search algorithm, and it was first published back in 1994 by Jimmy Yang et al. This was the introduction of sequest, the first algorithm of this type. In this case, we're going to be spending a lot of time talking about a sequence database, thus the database search method. So FASTA sequence databases were not actually created for proteomics. They were created for homology search, for recognizing which protein sequence has been seen previously in the sequence databases. Now, in this case, we're not using the FASTA algorithm, we're just using the sequence databases formatted in that way. And it's not much of a database, frankly. It has accession lines that say, this is the name of this protein sequence, and here's a short description about it. And then it has a bunch of lines of sequences themselves. Then it repeats. Here's the next name of a protein sequence. Here's a description. Here's another series of lines that represented sequences. So from the FASTA database, we were able to get protein sequences. Generally speaking, this is going to start with all of the proteins that are known for the organism of interest. So if you dealt with a human sample, you start with the human proteome. If you start with E. coli, there's very little point in searching human proteins against it. Okay, so these protein sequences in silico, in the software, need to be digested like they would have been with trypsin on the bench. So we need to emulate the behavior of trypsin in the software to create the peptide sequences that would result from those protein sequences. These peptide sequences then need to be expanded in some way. So I'm going to note that I'm mixing together information from the original publication of sequest in '94 and the follow-up version in 1995 that added post-translational modifications. PTM expansion allows us to ask which amino acids in these peptide sequences are allowed to be modified. The most common example of this is methionine. Because we allow methionine to appear in a lot of bench manipulations, it's exposed to oxygen rather a lot. And one of the things that methionine does, if exposed to oxygen, is to oxidize. So instead of having the normal mass of methionine, we have methionine plus 16 to reflect the gain of oxygen. So every time the software sees this methionine, it has to go this way, meaning not modified, or that way, representing modified. It causes a branch, essentially, in those peptide sequences. So having started with protein sequences, trypsin digested them to produce peptide sequences in silico. We then decorated them with post-translational modifications. Now this next stage is the really key insight that makes sequest work so well, which is that we don't have to compare every peptide sequence with every tandem mass spectrum. It would take a really long time to do that. And yet, that is the premise of the open search algorithm that's subsequently become quite popular. But in the original database search algorithms, we compared only those peptide sequences to tandem mass spectra that matched their intact masses very accurately. So if you have a peptide that has a mass of 1,000, it doesn't really make sense to compare it to a peptide sequence that would have a mass of 1,200. The mass spectrometer is able to tell those apart very, very well. Typically, a high-resolution mass measurement will know the mass of the peptide ion within 10 parts per million, something like 0.001 mass to charge. So we know very accurately what the masses of these peptides are. Therefore, we only need to compare peptide sequences that agree very, very well with what we saw when we measured that intact mass. But typically, this comparison is not taking place just on the basis of precursor masses. Instead, we need to be able to compare the fragments that should result from this peptide sequence to the ones that we saw in the tandem mass spectrum. So we are going to curate down this big list of decorated peptides to just those that match the precursor mass very, very well, perhaps within 20 parts per million. Those will be the candidate peptides that we compare to our tandem mass spectrum. For each of those, and only for those that meet the peptide mass filter criterion, we will predict the fragment ions that should result and compare them in a match scorer to the to the tandem mass spectrum. This last step, the match scorer, is the primary source of difference among different database search algorithms. We'll talk about just a couple of examples to explain why that's the case. But first, we would like to go into a little more detail on each of those steps. So let's start with the trypsin digestion step. You could imagine each protein as having a cutting site, for example, where every lysine and arginine is present. That reflects a trypsin digestion. So if each of these little slashes represent a potential place where trypsin might cut, you could imagine that the peptides we see are all going to be fully tryptic peptide, because trypsin only cuts where it is allowed to cut, and it shall cut every place it is allowed to cut. So fully tryptic peptides start at either a peptide N-terminus or a trypsin cut site, and they extend to either a trypsin cut site or the C-terminus of the protein. So these fully tryptic peptides then represent the best case scenario. Trypsin should produce this outcome under these circumstances. However, frequently we go looking for semi-tryptic peptides, and these can fall into two categories. The first is that they might start at a legitimate trypsin cutting site and then cut off somewhere we don't anticipate. That's this second example. Or they might be of the third example, where they start someplace we don't anticipate and they extend to a trypsin cutting site. So both of those are semi-tryptic peptides, but to search for them takes about 10 times as long, or at least it produces 10 times as many comparisons, as does a fully tryptic search. We must also consider the possibility that trypsin failed to cut somewhere we anticipated that it would. That's called a missed cleavage. So in this bottom example, we see that two consecutive peptides, fully tryptic peptides, remain glued together by a peptide bond, even though we expected trypsin to have cut between them. You need to specify how many missed cleavages are acceptable in your database search. And of course, the more missed cleavages you allow for, the more possibilities must be considered for each tandem mass spectrum. And also keep in mind, there are many regions of protein sequences that do not contain many arginine or lysine residues. As a result, those segments are very likely to remain undigested because there's no place for trypsin to have cut. As a result, we end up with these big blobs of protein sequence that don't produce very good-looking tandem mass spectra and thus are difficult to search for. All right, the peptide mass filter is, as I said, this very critical piece of the software that specifies, that contains the rules that say which peptide sequences can be compared to each tandem mass spectrum. And as I said, typically, when we run our database search of high-resolution mass measurements of the intact peptides, something that you get from like a Fourier transform instrument or from an Orbitrap, for example, would be that you allow for a plus or minus 10 ppm error on what that precursor mass could be. So the mass spectrometer may have measured something fractionally higher than what the true mass was, or it might have measured something fractionally lower than what the true mass was. Allowing for 10 ppm is a pretty narrow, very tight tolerance for allowing this search to proceed. And so long as your mass tolerance is big enough, you're going to identify lots of stuff. By keeping it nice and small, you're limiting the amount of comparisons that get made per spectrum. Now, in the bad old days particularly, we only had low-resolution measurements of the peptide ion. This is certainly the case if you were using something like a quadrupole ion trap, like an old LCQ or LTQ instrument, or today's Velos. Not the Orbitrap Velos, just the old the Velos without the Orbitrap. So in something like that, we typically had to allow for much more error in how much mass the intact peptide had than what was actually observed. So allowing for a deviation of up to 1.25 m/z on either side of that peptide ion is quite a lot more, allows for quite a lot more liberality in which peptide sequences get compared to each spectrum. But if you don't have a high resolution mass measurement, you're going to have to allow for more error in this mass measurement. Now, the time of flight style instruments, such as the triple TOF or something like an Agilent 6520, something like that, or for that matter, a Bruker Impact or a Bruker Maxis, can have very high mass accuracy under some circumstances, and under others may not. So I've written in that if you don't have mass correction that's applied to the masses of observed peaks in your mass spectra, you may need to allow for as high as 100 parts per million for a time of flight measurement. That does not really reflect the current generations of QTOF and TOF-TOF mass analyzers. If you're dealing with a very new type of instrument, you may end up using 10 ppm or 20 ppm just as if you were working with a Fourier transform instrument. Sometimes we just don't know until we've worked with that data type a fair bit to know what an appropriate mass measurement is. It's probably a good idea to read papers that have been published with your instrument type to know what is the typical kind of mass accuracy used in interpreting its data. We also want to be able to predict what the fragment ions should look like for a particular peptide sequence. This is something that might seem obvious to some, but might not be as much to others. So I would like to walk through this a bit slowly. Let's start with the case of a +2 peptide ion. That means that the peptide ion is carrying 2 protons on board, and when it breaks, we expect 1 proton to go with either side. So the B ion is going to be singly charged, and the Y ion is going to be singly charged. The software that we use for peptide for database search almost always assumes that when this peptide bond breaks, each is going to leave with 1 proton and thus be singly charged. We can then predict what the mass of a given B ion should be by adding together the residue masses of the amino acids contained within it and adding 1 to represent the hydrogen, or the proton rather, that's sitting on board, the hydrogen nucleus, okay. At the C-terminus, we have a very similar kind of computation. We have 3 amino acids here, we've got 3 amino acids here, but there's a really important distinction between what we see in the B ion and what we see in the Y ion. You'll note that the Y ion has a hydrogen stuck on its N-terminus and a hydroxyl stuck on its C-terminus. This reflects that the difference between a B ion of a given sequence and the Y ion of exactly the same sequence will be 18 apart, because 2 hydrogens and an oxygen, water, have a mass of 18 mass units. So even if a B ion and Y ion have identical sequences, there will be a mass difference of water between them. The other thing that we recall is that we're going to progressively be doing math on as the B ion series grows. So the B3 ion has the mass of these 3 amino acids and a hydrogen. The B4 ion is then going to add 1 more amino acid residue mass on top of the ones that were already in there. That's the difference between a B3 and the B4 ion. It's the mass of the amino acid at the 4th position in that peptide sequence. Okay, all pretty fair, I think, for the +2s, but let's talk about +3s. They actually get kind of difficult. First off, generally speaking, the peptides that can carry 3 protons are bigger, or at least more basic, gas phase basic, than the +2 ions are. So if you have a a pair of sequences that are both 12 amino acids long, the one that's got very few basic residues in it is likely to show up as a +2, but one that's carrying, say, 2 lysines and an arginine within it are going to be +3 instead. All right, so this +3 ion, we need to consider a pair of possibilities. Remember that in the +2, we were carrying 2 protons and 1 went with either fragment. In the case of a +3, we have 3 protons on board. So it might be that 1 proton leaves with the N-terminus to make a singly charged B ion, which means that 2 protons go away with the C-terminus, making it a doubly charged Y ion. It might, however, be the reverse of that. It might be that when this peptide breaks, the peptide ion breaks to produce a B ion that contains 2 protons, not just 1. As a result, the C-terminal ion, the Y ion, leaves with a single proton, thus becoming a singly charged Y ion. It is not easy to tell for every breakpoint in a long peptide which charge states the N-terminal B ion or the C-terminal Y ion will go away with. As a result, most database search algorithms use a model in which every N-terminal B ion is modeled as both a +1 and as a +2, and every Y ion is modeled as both a +1 and a +2. This means that for the same sequence appearing in +2 and +3 forms, the predicted spectra are twice as dense for the +3 variant as they would be for the +2 variant. So these are going to lead to very different kinds of scores, typically, for a +3 peptide than for a +2 peptide. The first type of score I'd like to talk about is probably the easiest type to understand, and it's the match counter. Probably the most widely used algorithm today that uses this system is MaxQuant. The Andromeda search engine within it is essentially building what we call a contingency table like this to determine the probability that this match could have taken place by random chance. This is a very typical kind of statistical model for it. So I want you to imagine that this tandem mass spectrum is now a jar of marbles, and everywhere that we observed a peak in the tandem mass spectrum is going to be represented by a black marble, and everywhere that we did not observe a peak in that spectrum instead has a white marble. Okay, so we have mostly white marbles along this mass spectrum axis, but a black marble reflects every location where a peak was observed at that mass to charge value. So when we do our assessment of a match, we're going to say of the M over Z values that we expected to be intense, which is to say at every location where we expected to find a peak given this sequence, how many of them contained peaks? So it might be that this particular peptide, when it breaks, should produce something like 20 different fragment ions. We're then going to inspect all of those 20 locations in the tandem mass spectrum and say, is there a peak there? Does it have a black marble rather than a white marble at that location? Some of those locations that we looked will in fact have a peak, and you want that to be a large number if this peptide sequence is a real match. But similarly, of all the mass to charge values that we expected to be intense, how many of them lacked peaks? So what if we go looking for a place where, say, the B2 ion or the B4 ion should have appeared, and there's no peak there, right? We're going to keep count of how many of those there are. Those 2 values go in the top of our contingency table. Now similarly, we have to ask about the locations where we did not expect to find a peak. So of the places in the M over Z spectrum that we expected to be empty, how many of them did have peaks? So what if you don't account for the biggest peak in the spectrum? That would be bad, right? So the software is able to say, of the locations that we expected to be empty, some of them do in fact contain peaks. We would like that number to be small if this peptide match is real. And of the mass to charge values that we expected to be empty, how many of them lacked peaks? Well, this might seem like a very silly thing to worry about if 90% of the locations in the spectrum do not contain peaks, and we expected almost all of those to not contain peaks. That number might seem to have very little value. But we need to contain all 4 of these possibilities. And these last 2 bullets correspond to the bottom of this contingency table. If you can populate a table with those 4 numbers, you can then compute an answer to say, what is the probability that by random chance we would have matched this many peaks or more for this spectrum and this peptide sequence. So we can compute that answer using what's called the binomial distribution. I believe that's the one used in Andromeda for MaxQuant, or by the hypergeometric distribution, which we've seen in other search engines. So these match counter engines can then say, what is the probability that by random chance we would have matched this many peaks or more for that spectrum? Okay, so that is a simple way to push good matches to the top and bad matches to the bottom. Now, the first one that was ever published in this space was Sequest. And I want to include it just because it shows up in a lot of algorithms, and it's a very different kind of answer to the same question, one that takes peak intensity much more explicitly into account. So MaxQuant does care how many peaks are above a particular threshold of intensity, but within that space it simply cares how many are above, how many are below that in intensity. Something above that is a peak, something below that is not a peak. Sequest's cross-correlation, on the other hand, takes intensity very much into account. It wants much more to match the intense peaks and to punish sequences that fail to explain big peaks, and that's what cross-correlation will do. I want you to, there's a lot of text on the screen, there's even a formula or two. I'm not going to ask you to worry about that for the moment. Let's instead say that we have the observed tandem mass spectrum, and we also have our model of what that tandem mass spectrum should look like for a given peptide sequence. Now, you can imagine that if the model we have is for the correct sequence, we're going to get a very good correspondence where the peaks are intense in the observed spectrum, they are also intense in our predicted spectrum, and we see this great alignment between the two. And we can compute the sum of intensity products across that as a way to evaluate the quality of this matching. But I also want you to think about the possibility of scooting the observed and predicted spectra against each other by some range up to 75 Daltons either way. So let's say we set our observed and predicted spectra out of phase with each other by 1 mass to charge, or 2 mass to charge, or 3 mass to charge. What you see in this spiky hairball that appeared back in the 1994 paper is that when you have a correct sequence being considered, at tau 0, at no offset between the two, you get this very high spike. And as you move to greater and greater shifts away from each other, you get this kind of hairball above it. So the cross-correlation score that we report in Sequest reflects how tall this big spike is versus this random matching as we move these two sequences with respect to each other. You want this spike to stand out way high against everything else. This search score is going to need to play 2 very important roles, one that is per spectrum and another one that's across the entire data set. First off, we want to boost the correct sequence to a very low rank, to the best position, to the top rank of this long list of peptide sequences that are getting compared to this spectrum. Generally speaking, if the database search algorithm does not push the correct sequence all the way to the top for a spectrum, there's no way that this spectrum is going to be correctly identified by the later steps. That's not true for all applications. Systems like Percolator, for example, can do re-ranking after the fact, but generally speaking, most of the software frameworks we use expect that the database search engine must get the right answer to the top, or all is lost for the spectrum. But we will also want to filter peptide spectrum matches across many spectra. So we're going to place greater confidence in tandem mass spectra that have very good scores, and less confidence in spectra that have very poor scores, which means that we need to have well-calibrated scores. We need to be able to create scoring systems that allow us to compare among different spectra that share some characteristics, for example, they're all +2s, to be able to say that the score from this PSM, representing this spectrum, is comparable to the score for this PSM, representing an altogether different spectrum. That's the property of well-calibrated. Lots of takeaways. Database search asks which peptide from a sequence database drawn from a FASTA database is the best explanation for this tandem mass spectrum. Rather than inferring sequences from tandem mass spectra, we are dependent on the sequences in the database to tell us which peptides are possible, and only those are going to be have the spectra predicted for them and compared to this empirical set that we have from the observed tandem mass spectra. And importantly, we need to consider what are the failure scenarios for this? What are the properties that would cause us to fail to identify this tandem mass spectrum successfully? It might be that the tandem mass spectrum is simply too low in its signal to noise. It might be that there's just a tiny amount of that peptide present, thus we didn't get a good tandem mass spectrum from it. It might be that the sequence that represents this tandem mass spectrum is simply not in the sequence database. If you're searching a human sequence database and you didn't add bovine or porcine trypsin, peptides from bovine or porcine trypsin would not be identified because they're different sequences from anything in the human sequence database. It might be that you have some sort of non-specific proteolysis going on. A lot of biofluids have a little bit of protease activity in them. Was it inactivated? Did you have something like Zorching going on, where the peptides coming into the electrospray ion source were being blown apart by too energetic a source? That can lead to what look like non-specific proteolysis events. Or it might be that you had a very inaccurately measured peptide mass. As a result, the correct sequence fell outside the mass tolerance for the peptide mass filter. There are other reasons too. So this technology is not bulletproof. Not every tandem mass spectrum is going to have its correct sequence on the top of the list. And that means we're going to need some way to assess error, which is the next segment of this video. Take a moment to pause, refresh yourself, and come on back. Welcome to part 3, or part C, of our Proteome Informatics series. In this part, we'll talk about how we can control error through target-decoy search. So let's start with a few definitions. We want to do false discovery rate control. To say, we want to be able to say what fraction of peptide spectrum matches in a given set are estimated to be false. Okay, so we're trying to allow for a little bit of wiggle room. Rather than say, this set of peptides is absolutely certain, every last one of these is sure to be present, we're trying to allow ourselves a little bit of uncertainty and say perhaps 1% or 2% of these peptide spectrum matches are not correct. This allows us a lot greater sensitivity in detecting peptide sequences. So the typical way that we do this is target-decoy analysis, which means that when we do our database search, we're going to provide sequences that are real, to reflect, say, the real human proteome, but we're also going to include some number of sequences that are decoys, which is to say they are sequences that do not exist in nature and have not been identified. Because every time we identify one of these known fake sequences, we can say to ourselves, gosh, I bet another one is hidden in here that looks like it's a real sequence, but is actually falsely identified. So our sequence database needs to have equal numbers of target and decoy sequences. If you have 70,000 human proteins in your sequence database, you will also want 70,000 fake proteins that are very similar in sequence composition in terms of which amino acids they contain as the human sequence does. The most common way that people implement target decoy search is that each protein sequence in the human proteome is given in its normal orientation, N-terminus to C-terminus, but it's also shown in the reverse orientation, where the sequence is reported backward. So this reversal means that every time we identify a peptide to the a known fake sequence, we assume another one is hiding in there that looks like a real sequence, but it's a fake hit. Okay, so the hypothesis behind this is that a false identification, a spectrum that's matched to the wrong sequence, has just as good a shot of matching to a target sequence, a real natural sequence, as it does to a decoy one, to a reversed sequence. Given that if we imagine all of our PSMs stacked from best score to the worst down on the floor, we're going to try to find some place that we can apply a threshold against this list of PSMs to say everything above this is accepted as a confident hit with some estimated error, and everything else we are throwing out. They're all junk. Okay, so the way that we find where to set that threshold is to say, for a given threshold, what is the number of matches above it that we estimate to be false? So in this case, given that the decoys are known to be false and an equal number of target sequences are projected to be false, we simply double the number of decoys above this threshold. The math varies a little bit if you throw the decoys out, but not tremendously. All right, so we can then say, given that we now have an estimate of how many of these PSMs are false above this threshold by doubling the number of decoys, we can then divide by the number of matches, the number of PSMs above that threshold, and say this fraction of all of the matches in this set is expected to be false based on target decoy analysis. Okay, there are several terms of art that show up in this. We've already mentioned the reversed versus randomized database difference, but here we have a concatenated target decoy search and a separate target decoy search. So in concatenated, we have a single sequence database that contains both real sequences and fake sequences. All of the fake sequences have some sort of name in their accessions to indicate to the downstream software this is a decoy. Sometimes it's XXX, sometimes it's REV, sometimes it's decoy. You find all kinds of different uses for this. But in this way, the sequence database contains each protein sequence in two forms, essentially. However, a lot of software frameworks, such as those built around Percolator, I believe, separate the search. You do one search against the target sequences, a second search against the decoy database, and then you have two different lists of PSMs to play off of each other. Okay, so concatenated versus separate target decoy. I would like to try to explain how this works kind of visually. So I decided to simulate a data set. We have 10,000 correct PSMs that have an average score of 6 and a standard deviation of 1. That's represented by this hump over here. And we have a set of incorrect PSMs that have a score that's lower typically. So we had an average of 6 for the correct ones and an average of just 3 for the incorrect ones. But again, a standard deviation of 1. This is a fake score. It's just a model to help us understand it. Ideally, a great search engine is going to push the correct sequences to really, really high scores and all the incorrect ones to very, very low scores. But they're all going to end up with some amount of overlap. That's the area we really want to dig into. So we can see that the incorrect PSMs are divided into 5,000 decoys and 5,000 target hits. All right, that's why we see both red and blue cresting down here, because there are both incorrect decoy hits and incorrect target hits. So where would we have to place our threshold here? Where would we set our line so that we had exactly 0 false matches? You probably thought for a moment to yourself and said, you know, if I want to eliminate all false matches, I'm going to need to set my threshold higher than the best scoring decoy. So I'm going to look at where all these reds are. Okay, still reds, still reds, still reds. Here, right here it is. This is the end of the red distribution. I can set my threshold right there, and everything above it I can estimate to be correctly identified. But look at the cost. If you set your threshold all the way up here, you're throwing out about half of the correct peptide spectrum matches. that is a big hit to your sensitivity. Because I got to tell you, every biologist you work with is going to want to have more peptide spectrum matches, more proteins identified. And if you place your criterion so high as to exclude all decoy hits, yes, you can have great confidence in your hits, but there's a lot you've left on the table that you would have liked to accept as well. So, how much error would we have if we instead placed our threshold at the valley between the peaks? So right here, we have this little dip between the two. I can select that as my threshold. Then I can ask what fraction of the matches above that line are decoy hits. But remember, for every bit of red that appears in this region, I must assume there's a matching false hit among the targets. So I've got to double this red fraction above that line to get the estimate of how many total are erroneous. Okay. Oh, and I should have mentioned that in this case, the blue false hits are layered on top of the red false hits here. Okay. Now, intuitively, this might make sense to some folks, but not to others. So I want to walk through this a different way. I've now sorted all of my PSMs in a giant spreadsheet. And every time I had a target hit, I'm coloring it blue, and I'm leaving it uncolored if I had a decoy. But you can imagine that those are red if you like. Here we have two different regions of my big spreadsheet. This is PSM 6676 to 6698, and over here we have PSMs 8306 and 8328. So the closer they are to the top of the list, the more likely they are to be target hits. So if I looked at the very first page of these, they would all be blue. When I look at this region, I can see that hit number 6681 is a decoy, and hit number 6692 is a decoy hit as well. But out of this entire block of PSMs, just two are decoys. But the enrichment in decoy sequences is going to get worse and worse the lower down our list of PSMs that we go. So by the time we get down to this block of PSMs, we see that 8308 is a decoy, 8312 is a decoy, 8319, 8320, 8324, and 8328 are all decoys. So we have 1, 2, 3, 4, 5, 6. We have 3 times as many decoys here as we do in the left pane, which means that including all of these comes at a cost of 2 decoys. Including all of these has a cost of 6 decoys. So finding the right place to set this threshold is going to be a question of how much damage, how many decoys are we willing to accrue in order to get better sensitivity. Another way to say this is that to achieve more sensitivity will come at the cost of a higher error rate. Here we're showing a statistic called the Q value, which is to say, what would the best error rate be if we were to accept a given peptide spectrum match? What we see is that up at high scores around 7, we're over the mean score of the true distribution. We see that there are a vanishingly small number of errors. Once we get to about 6, though, which was the mean value for correct scores, we see that we're accumulating some number of decoys. But this keeps getting worse and worse and worse, and by the time we hit a threshold of 4, we have a pretty high error rate that adding new PSMs is coming at an expected error rate overall of 14%. So finding a place where we can allow for less error is the way of finding basically how tolerant we are of errors determines what sensitivity we can possibly achieve. They come at counter purposes here. The more sensitivity you demand, the worse your specificity gets. The better your specificity gets, the worse your sensitivity. So there are several assumptions that play into this. We've already mentioned some, but I guess we started with this one, that when a peptide spectrum match is falsely identified, it has the same probability of matching a decoy as it does a target. That's one we mentioned a few slides ago. But keep in mind that a given peptide could potentially appear in both target and decoy sequences. The most likely scenario for that is that you have a peptide of, say, 4 amino acids. A 4 amino acid peptide has a pretty good chance of appearing in both natural sequences and in either reversed or randomized sequences used as decoys. So we need to be careful that a very, very small number of peptides appear in both target and decoy sequences. So short peptides frequently appear in both targets and decoys. Multi-pass search is something that we haven't talked about previously, but imagine that you cut down the search space of a second round of database search. Maybe you do a fully tryptic search first to say, these 3,000 proteins out of the whole proteome are the ones that are actually detected in this sample. I'm now going to dig into those very heavily and consider things like sequence variants and post-translational modifications. That's multi-pass search. And it may be that the way we implement multi-pass search can violate this second assumption, that people may include too few decoys for us to detect to set the error rate correctly. The other thing is that our estimates of false discovery rate at very low levels tend to be very noisy. That if you're trying to control peptide spectrum match to, say, a 0.0001 error rate, that number will bounce around quite a lot from raw file to raw file, from LCMS experiment to LCMS experiment. So our ability to control error at that very low rate can be rather noisy when we try to find these thresholds. A fair number of takeaways again. The target decoy technique is now a standard tool for controlling false discovery rate in proteomic identifications. But I've been spending all of this time talking about aggregate error, to say, for this collection of IDs, what fraction of them are false. That is not the same thing as trying to say, what is the error of the chance of error for this particular tandem mass spectrum? It's quite a different application, and some types of applications, such as post-translational modification searches, really benefit from being able to say, what is the probability of error for this PSM? All right, so aggregate error is not the same thing as aggregate peptide error. Oh, that's even a different question. So the error rate we have for spectra that have been matched to sequences may be different than the error for peptide sequences. If you're asking of all of the different peptide sequences we observed, what fraction of them are bogus, that's different than saying what fraction of the spectra were bogus hits. And the aggregate error for peptides is not the same thing as the aggregate error for proteins. That can escalate rather surprisingly. Tools such as the transproteomic pipeline, which is very widely used in things like Philosopher and in Scaffold, and tools like Percolator, which I've mentioned en route, frequently use decoy hits in a rather different way than what I've described here. This is the target decoy approach I've described here is kind of the simplest way to apply these data. But the systems like the TPP and Percolator will instead do some sort of either a distribution-based analysis or a machine learning analysis to determine how to leverage that information about error. Okay, take a little break. When we come back, we will talk about protein identification, more accurately, protein inference. See you in a minute. Welcome to segment E. You've made it all the way to protein inference. Well done, you're halfway through the whole six segment section. We're going to talk now about inferring proteins from peptide IDs, or from peptide data. Let us start with the idea that when we identify a tandem mass spectrum, that tandem mass spectrum gets matched directly to a peptide sequence. So when we talk about identifying a tandem mass spectrum, it is identifying a peptide. The protein lists that we build from shotgun proteomics are inferred, which is to say that we have the presence of peptides, and these different peptides can be grouped together in ways that allow us to infer a protein sequence that explains all of those peptides. So our tandem mass spectra get matched to lots of different peptide sequences. And yet this proteolysis that we're relying upon on the bench, the use of trypsin, is resulting in a loss of connectivity among peptides. Just to state something very obvious, I think, when we identify a particular tandem mass spectrum to a given peptide sequence, there's no real reason to expect that the next tandem mass spectrum we see is from the same protein, just a different peptide from the same protein. The peptides are all being separated by hydrophobicity, and that means that the different peptides for a given protein are all over that gradient. Some are relatively hydrophilic, and thus they come off the column very early, and some of them are very hydrophobic, and they come off the column very late. So the connectivity among these tandem mass spectra that tells us that these all come from the same protein is something we don't generally have in solution digests. If you need something like that, you should probably consider something like in-gel digest, which means that you're separating the proteins by gel, and then you're digesting just the proteins at this spot into tandem mass spectra. That preserves the connectivity among them. Okay, so proteolysis is the source by which we're losing this connectivity. We infer a set of proteins that will account for the peptides we have observed. If you are interested in keeping this connectivity for real, you should probably consider top-down proteomics. I've included a little logo here from the Coalition for Top-Down Proteomics. It's a growing body of people who are working with tandem mass spectrometry of intact proteoforms. It's a very valuable way to go after exactly which amino acids are connected to which others in the polypeptide. So that set of technologies depends on proteomes collected without a digestion step. All right. So many different peptides may appear in multiple protein sequences in the database. This might be obvious, but again, there are several different reasons why a given peptide shows up at multiple places in the protein database. First off, it might be that you're using a multi-protein database. Maybe you're using the UniProt database, for example. UniProt has proteins for a bazillion different species in it. If you're searching that, you might find that a peptide, for example, for actin in your species is identical to that peptide for actin in other species. We call this orthology, that we have different proteins from different species that share commonalities in their sequences. All right, it might also be that you're working in a species that has undergone gene duplication at some point in its evolutionary history. Humans certainly have. So when we look at gene duplication events, we see that the different duplicates of those genes have been drifting apart from each other in evolutionary time, but they still contain some sequences that are preserved perfectly between them. That's paralogy or paralogous sequences. But the most common reason why you're going to have the same peptide sequence appearing multiple times in your database comes from isoforms, which is to say that a given gene produces multiple messenger RNAs, and those different messenger RNAs have similar overlapping translations. As a result, a given peptide from one isoform is likely to appear in other isoforms from that same gene. So isoforming is the third reason why we will frequently see, at least in a human database, and in all of its eukaryotic kin, some amount of duplication for the same peptide appearing in multiple proteins in the database. Now, I would like to talk about the bad old days, and I would note that I've been in the field of proteomics since 1996. And back in the late 90s, things were running amok. They were not going well, because a lot of people would look at exactly the same data and come up with completely different visions of which protein sequences had been successfully identified. The maximal list is one of the canonical terms we use to describe why the bad old days were so bad, which is to say that people would frequently claim that if a peptide were identified or were matchable to this protein, you could claim that protein existed in your sample. that's naturally a little dubious for the reasons that I've just described. So a maximal list is the set of proteins that include any observed peptide in your experiment. That is clearly too big a list to publish today. In the late 1990s, things had gotten to such an impasse that journal editors in the field of proteomics got together and put together sets of rules about what kinds of criteria you had to reach in order to claim a given protein was identified. And things like the presence of isoforms in your database really requires a lot of justification to say that this isoform rather than another is present in your database. We're going to talk about how some of these rules play out as we move ahead. But first, I would like to introduce you to the term parsimony. Parsimony. Now, a long, long time ago, I was a little boy, and when my parents went to the shopping center, my brother and I would wander around the toy section looking for anything that might catch our eye. I was really interested in buying a Saturn V rocket in plastic that had like a spring-loaded capsule release thing on it and everything. I thought it was the coolest thing ever, and I wanted to take it home. But you know what? I looked at its price tag and I said, that's just too expensive. It's too expensive. And my brother would say something along the lines of, Dave, you gotta live a little. You're cheap. You're cheap. Just pay the money and get it. Now, as a little brother, it was hard to hear that I was cheap. But I gotta say, I'm still a cheap, thrifty guy. I like not spending money wherever I can avoid it. Now, as a child, being called cheap hurts. But as a scientist, being called parsimonious is positively a great thing. What it means is that you're very careful in weaving explanations to explain your data. It means not going further than your data can actually take you. So I didn't want to spend my money as a child on something that was just going to break. And in the same way, you as a scientist should be very careful in hypothesizing the presence of a protein if you're not actually sure your data can support that declaration that this particular protein and no other is present in your sample. This concept of parsimony was first stated in Western culture, at least, by William of Ockham, who said, "Plurality ought never be posed without necessity," which means you shouldn't claim lots of things if you don't absolutely have to. Now, he did not actually speak modern English. Instead, he wrote this in Latin. So, pluralitas non exponenda sine necessitate. That was how he wrote it back in the 14th century. So, remind yourself that as a scientist, you never want to go beyond what your data can actually support. Let us talk about how this plays out. Let's start with indiscernible or equivalent proteins. They go by both names, and different software frameworks can call them different things. Let us imagine that protein A and protein B could each be used to explain the presence of peptide 1, 2, or 3. Protein A could be why peptide 1, 2, and 3 are seen, or protein B could explain why peptide 1, 2, or 3 are present. And we have no additional peptides seen for either protein A or protein B. As a result, we should say, when we analyze this in our protein inference software, that either protein A or protein B could be the explanation for why we see peptides 1, 2, and 3. So typically, when we're saying how many proteins did we identify, we group these together and say this single protein group accounts for these 3 peptides. Okay? That's a pretty straightforward example, I hope. Let's now move to another example. It's quite similar, but has a very different outcome. In this case, protein A can account for peptides 1, 2, and 3. It'll account for all 3 of them. That's great. Protein B could account for peptides 2 and 3 only. So do we claim that both proteins exist, or do we claim that just protein A is present? In this case, the concept of parsimony says that protein A can account for all 3 of these peptides, but protein B could only account for 2 of them. Because of this, protein B is relying on a subset of the evidence for protein A. As a result, most software today will not even tell you that protein B was a possibility, because protein A is a superior explanation. It accounts for more of your data than did protein B. And once you've claimed protein A's existence, there's no longer any justification remaining for claiming separately the existence of protein B. But let me ask you a more challenging question. Does that mean protein B is not present? Have we proven protein B is not present? No way. No way. We do not prove that proteins are absent by shotgun tandem mass spectrometry. We always assume that there's some set of proteins that are present that we just didn't have enough sensitivity to reach. So it's possible protein B was there. And in fact, if we do another experiment, we might just randomly, even if we're using exactly the same samples, we might this time detect a peptide that could only be explained by protein B. And it would look pretty silly, wouldn't it, to say we've claimed protein B was not present. Okay, so B is typically stripped away, is hidden from us, because protein A explains more of the data than protein B does. But things can get even a little weirder than that. In the case of subsumable proteins, we have a case where the data for a given protein is not the subset of that for just one other protein. In this case, peptides 1 through 4 can be explained by just protein A and protein C. And protein B, which explains a little bit of the evidence for each of those two other proteins, is treated as subsumable, which is to say that all of the evidence for protein B, it can be accounted for by a combination of other proteins. So it's treated just like subset proteins. It gets stripped out, and typically just proteins A and C will be listed. These will be unrelated in this case because protein B is no longer there to link them together. So let's imagine a scenario there, though, where your software starts by claiming that protein B is present. Now it needs to invoke proteins A and C in order to explain peptides 1 and 4. So some software fails to handle subsumable proteins satisfactorily and claims 3 proteins are here by accident rather than limiting itself to just proteins A and C. Subsumable situations can lead to great complexity in trying to say which protein should be claimed first. Sometimes these situations get very badly out of hand. My friend Carl Klauser over at Broad Institute provided some really great data from something called a xenograft. In this case, we had a human breast cancer tissue that was injected into mice, and the mice then developed tumors. But those tumors contain both human cells that are proliferating rapidly inside the mouse, along with some mouse material itself. So there's a mix of mouse protein and human protein, as well as isoforms and gene duplicates that exist within these species. So we see that in this case I've created a bipartite graph where everything on the left is a protein, everything on the right is a peptide, and these lines indicate which proteins can be justified based on which peptides at the right. So those arrows are indications that this protein can explain those peptides. How many different proteins do we need to invoke to explain this set of peptides in this case? The answer, surprisingly, is not all 7 of these blocks, but rather just these 2. Just those 2 blue boxes have an arrow to every single one of the peptides at right in combination. So by claiming just those 2 proteins, we can account for all of the peptides. Therefore, the other 5 groups of proteins, we don't need to pay any attention to at all. So this type of inference of proteins becomes rather complex when we have a complex mixture of organisms in one sample. Small changes in how we do our filtering can make a very big difference in how many proteins we claim for a sample. In each of these rows, I'm going to use exactly the same sets of peptide spectrum matches, but I'm going to filter them very slightly differently. In each case, I'm applying a peptide spectrum match false discovery rate of 3%, which is to say that 3% of all of the PSMs are false. But I'm going to apply another rule on top of that asks how many peptide spectrum matches must I have for a protein before I will call it identified, and I'm using parsimony in every case. Let's start at the worst case. If I start with just a 2-peptide rule, in other words, I'm saying only 2 peptide spectrum matches need to be present for a protein for me to claim it's been identified. I have 252,000 confident peptide spectrum matches. They match to about 103,000 different peptide sequences, and there are 8,342 protein groups. So at that, my biological collaborator is really excited. Wow, I get this huge number of proteins with this experiment. I'm fantastically happy. But something is lurking in the data. Even though I allowed for a 3% peptide spectrum match false discovery rate, the protein false discovery rate is 8.9%. Can my collaborator publish a list of proteins that has a false discovery rate of 8.9%? Not happily. A lot of peer reviewers today will bite your head off for something like that. You really want that number to be less than 5%, and frankly, a lot of reviewers would really prefer that it's below 1%. So if we change our rules very slightly, now instead of requiring just 2 peptide spectrum matches per protein, we require 3 peptide spectrum matches per protein. The number of peptide spectrum matches falls very, very little, 252,000 to 250,500. The number of peptides falls from about 103,000 to about 101,000. The number of proteins, however, changed hugely. About 1,000 proteins fall off of our list in just going from a requirement of 2 PSMs to 3 PSMs per protein. And now we see that instead of having an 8.9% protein FDR, we have a 3.3% FDR. That might be satisfactory, or it might not. Depending on what kind of peer reviewers you face, you might need to be even more restrictive. If you move up to 4 PSMs or 5 PSMs required per protein, you can see that the number of PSMs continues to fall, but not a lot. 252,000 down to 247,000 in the worst case. The number of peptides that we see falls some, but not a lot, from 103,000 to 98,000. But the number of proteins that we claim has dropped all the way from 8.3 thousand to 6.4 thousand. It's a huge fall. And yet the protein false discovery rate got a lot better. So we frequently see that very small perturbations in how we filter peptide spectrum matches lead to huge changes in how many different proteins we claim are present in our data. So again, our takeaways are that the process of protein inference is complicated by the homology among species and within a single genome, as well as isoforms, which are, in my view, the big issue in eukaryotic proteomics. Reporting a large number of proteins can seem impressive, but the reality is that the protein counts that we produce from bottom-up proteomes are highly subjective. The software could have a very different number of proteins claimed by very small changes. And frequently, people don't even tell you what the empirical protein false discovery rate is for a very large collection of proteins. And when you check it by looking at their spreadsheets, you discover it's much, much worse than what any peer reviewer really should have accepted. Parsimony is a conservative rule to guide minimal protein lists, to guide us in making minimal claims about our experiments. But it may strip away genuine hits. There may be proteins that really are there, but because they are subset proteins or subsumable proteins based on the evidence we've acquired, we allow them to fall away. So a lot of people have argued that this is far too conservative an approach to take in proteomics, but this is largely the set of people who want to have a longer list of proteins. From a statistical point of view, from a bioinformatic point of view, I do not like going any further than what our data can sustain. And if you don't identify peptides that tell you that this particular protein group must be present, I really feel like it's dangerous to claim otherwise. Okay, so that's an old fuddy-duddy's appraisal of how we should infer proteins. I hope it's useful to you, because at this point, segments E and F are moving in a really different direction. they're going to start talking about the fun of quantification in shotgun proteomics. So hold on to your hat, it's going to be a wild ride. Hi, and welcome back. We are ready for segment E of this 6-part series that starts us in a rather different direction. Previously, we've been talking about identification of tandem mass spectra of peptides, and from that, the inference of proteins. Next, though, we're going to talk about the extent to which we can quantify from proteomics data sets. In this part, we're going to talk a little bit about what's been done in attempting to quantify proteins on the basis of label-free data sets, which is to say that we're just dealing with samples as they are without any sort of isotopic enrichment or isobaric labels. We'll get into those topics in the next segment. But for now, I'd like to talk about something, kind of the most basic aspect of what you can get from shotgun data in terms of quantification, with the discussion of spectral counting and chromatogram integration. Spectral counting is a method that was published all the way back in 2004 and has been used quite frequently in the label-free shotgun proteomics world. It is a relatively simple technique, and yet I find that there are a lot of people who misunderstand several aspects of it. So I will start by saying, at first principles, a given protein will match to more spectra when it is concentrated than when it is not, if the proteomic background is largely unchanged. So you might imagine that you have a bunch of serum samples where just a few proteins are changing in the disease cohort versus the control cohort. It should be reasonable to think that the serum proteome as a background is not changing all that much, even though individual proteins may be popping up and popping down. Now, I will say this of biofluids, there are a lot of special rules that come into play, so using them as a place for proteomic discovery, biomarker discovery, is fraught with a huge number of issues. I will not be getting into those here. But generally speaking, if the background of proteins remains largely unchanged and a particular subset of proteins is rising and falling from between a control and a case cohort, then we would hope to be able to recognize that through spectral counting. The result is frequently described as semi-quantitative. We can detect that a change has happened, and we can detect the direction of that change. But quantifying it exactly to say that this protein rises exactly 40% in cases above what it does in controls is a level of precision beyond which we would hesitate to go. That being able to say that this protein changes upward in the cases versus the controls is a whole lot easier statement to make than to say that this changes between 35 and 45%, for example. Okay, and a very key aspect of this that must not be ignored is that we do not compare the number of spectra observed for one protein to the number of spectra observed for an altogether different protein. That is, this way madness lies. Essentially, you can compare the counts observed for A100 here and A100 there, but you don't compare A100 here to the number of counts seen for albumin. That's never going to go well. So, in short, we expect to see that as the number of copies of protein increase, the number of spectra observed for them will also change. So why does this work? Why would we find that the number of spectra observed for a protein is changing? There are a lot of people who will say that under so long as we use dynamic exclusion, a common feature in thermomass spectrometry datasets, to prevent the same peptides from being matched to spectra multiple or produced in multiple spectra, to try to suppress that influence, that spectral counting couldn't work. But we're not really talking about the number of times an individual peptide is being measured in a tandem mass spectrum. Instead, we're saying that the set of peptides observed for a protein increases when that protein is present in greater abundance than in another case. So let's start with this basic principle that every proteomic sample contains far more peptides than the mass spectrometer can sample comprehensively in tandem mass spectra. That might seem a little counterintuitive. If you run a single protein that's been digested in a tandem mass spectrometer, aren't you going to see the same peptides every time? The answer is no, you're not. And this is a surprising feature for a lot of people because they assume that there is such a thing as a defined mixture of peptides derived from proteolysis. And the reality is that mass spectrometry is a sampling process. Mass spectrometry is going to try to produce tandem mass spectra from as many of the peptide ions as are available, and yet you don't have any guarantee that all peptide ions will be sampled in tandem mass spectra that can be successfully identified. There are a lot of things that can alter very dramatically which spectra are observed when. Okay, so let's just start with something basic. If you were to use trypsin on different samples, trypsin would sometimes cut a protein to completion and other times it wouldn't. You can think of each site in a protein as being as having some probability that it will be cleaved under ordinary denaturing and reduced conditions. And yet, this probabilistic evaluation of how a protein gets digested has a weird implication that some peptides for a given protein are observed with very high probability and other peptides are produced with very low probability. Consider a peptide that's got 3 internal cleavage sites where trypsin should cut, but each of these has a different probability of cutting associated with it. Occasionally, you might see what is essentially 4 peptides all glommed together because they were never digested in the first place. But sometimes you're going to get a fragment that contains 3 of those regions or 2 of those regions or just the fully tryptic peptides themselves. The probability of cutting at each of these sites leads to some peptides being obviously visible for a protein and some being very rarely observed for the same protein. Okay, a peptide must compete with others for ionizing protons in nanoelectrospray ionization. In order to detect a peptide at all in the mass spec, we must ionize it. And there are only so many protons to go around. You may have something like signal suppression where one peptide is so much more dominant in a sample that it's grabbing all the protons that are available. And if that peptide comes off with one you're actually trying to detect, you might find that you simply have very little signal observable in the mass spectrum for your peptide of interest. Similarly, just because you generate the tandem mass spectrum does not mean that it will necessarily be identified. Each tandem mass spectrum has some probability with which it would be identified successfully in database search. At some level, when you have just a tiny amount of that of the fragment ions to measure, the database search may fail, and so you don't see that peptide even though it was sampled in a tandem mass spectrum. So think of this process of sampling tandem mass spectra and identifying them as a competitive process. The peptides are competing for identification. The peptides that come from abundant proteins have an advantage over those that come from very rare proteins. A lot of times, the data that come from spectral counting experiments look something like what you see on screen right now. In this case, we're looking at real data. These were produced in 2008 for a 2009 study of the Association for Biomolecular Resource Facilities. In this case, we had 5 shots, 5 LC-MS experiments representing red E. coli. These are technical replicates rather than biological. And over the right, we have 5 shots from yellow E. coli. Now, I don't need to get into what differs between the red and yellow cohort here, but we sent participants copies of the raw files for these 10 experiments and asked them which proteins changed. A lot of the teams made use of spectral counting, and other teams made use of what the technique we're going to get to next, which is integrating chromatograms. But the data looks something like this. If you see a particular protein, you can see how many spectra were observed or were matched to peptides of that protein as you go across the row. So when you see something like P0A65F5 here in about the middle of the table, you see that it ranged from 66 spectra up to 80 in these 5 technical replicates. Over at the right, we see that it ranged from 20 to 26 spectra in yellow. So you might just look at this to say, well, gosh, every experiment in red saw more spectra for this protein than each experiment in yellow. That's a pretty good case. What you're performing there is called a U-test, to evaluate what the range of values from one cohort versus the range of values in the other. And you could get a significant p-value for this protein under those circumstances. But data like these illustrate that there's quite a lot of bounce we see in these numbers. If there's variation in the number of spectra we see for a protein across technical replicates, think about how much worse this gets when we're dealing with biologically different replicates rather than just technical reshooting the same sample. All right, so CPTAC, which was a big project in clinical proteomics from the National Cancer Institute in the United States, launched a big reproducibility assessment to try to evaluate the role that proteomics could play in cancer biomarker research. So we started with some fairly basic samples, a bunch of yeast lysates. What can you learn about our technology from yeast lysates? A lot. In this case, we were able to look at 3 different ion trap instruments, LTQs made by Thermo, and 3 different Orbitrap instruments, also produced by Thermo. These were run by different operators using exactly the same samples that were digested up at NIST and sent in peptide form to each of the participating labs. In the first case, we see that the first shot of yeast, we have essentially 100% of the peptides seen in one run. Then when we run a second or a third or a fourth or a fifth or a sixth replicate, we ask how much did the number of peptides observed change as we gained these second and third and fourth experiments. So you can see that for the LTQ, having shot the same sample a 6th time, you were still identifying new peptides that hadn't been seen in the first 5 shots of yeast. The same is also true for the Orbitrap instruments, although it appeared that the number of peptides we gained by reshooting a sample in the LTQ was continuing to rise more quickly than for the Orbitrap instruments. What do we expect as a typical amount of variation in the number of spectra we observe for a given protein? Again, over here we're looking at just yeast, and we're asking from a statistical point of view, what is the typical amount of variation that we observed in these? And in this case, we see that for the 3 LTQs, these first 3 rows show a fair bit of variation. Again, for the top protein, a range from 18 spectra up to 29 seen for this protein. In the second case, we see that 4 of the 6 experiments identified exactly 2 spectra for this protein. The last 2 both produced more. Stuff like this is going to happen because of random effects. If you were to be looking for a biomarker and replicates 5 and 6 represented cases and the other 4 replicates represented controls, you might be tempted to say, well gosh, it's always higher in cases than in controls. But in this case, we know it's a completely spurious amount of variation. This is a typical amount of variation to expect for proteins being sampled repeatedly through LC-MS-MS experiments. All right, so let's move away from spectral counts because there's obviously a lot of bounce there and move to something like chromatograms. Chromatograms are definitely a favorite approach for people to use in analyzing label-free proteomes. And I think the best example of this is the MaxQuant software. We'll be talking about that in just a minute. All right, so liquid chromatography is separating peptides to different retention times across minutes or hours. So typically we think of an hour-long experiment or 90-minute experiment having either 40 minutes or maybe an hour of a slow gradient of hydrophobicity slowly increasing, and so the hydrophobic peptides come off last and the hydrophilic proteins come off first. As a result, a given peptide is going to show up at some moment in time as it starts to elute off the column and then slowly fall back down again at the trailing end, essentially, of its chromatography. So you generally would have something like 30 seconds for a typical peptide in one of these hour-long experiments. So you have only 30 seconds during which this peptide is eluting to capture a tandem mass spectrum of it and thus identify it. But here we're talking about quantitation, and the quantitation is taking place in the label-free context at the level of the MS scan. So we have the moment when that peptide first appears, and then it tapers off and falls back to Earth again. So if that all takes place within 30 seconds, we have to ask, how are the MS scans spaced across that chromatogram? If you've got just 3 mass spectra collected during this interval of 30 seconds, your amount of measurement on the intact peptide ion comes in just 3 shots, and that's not a very good way to estimate how much of this thing there is. Typically, we're going to want to put together a chromatographic peak area to tell us about this spectrum. We'll talk about that a little bit later, but for now, what I want you to keep in mind is that the extracted ion chromatogram that we see for a particular peptide represents its rise and fall in signal intensity across MS scans that are collected during its moments of appearance and disappearance. Okay, so MaxQuant makes use of this chromatographic peak area to compare how much a peptide has changed across different experiments. And given that we usually have multiple peptides identified for a protein of interest, we can ask how these peptide areas compare for all peptides of a given protein to give us kind of an estimate of what's the high and the low of how much this peptide is changing. But I'd like you to think about these peaks as they appear in mass spectra themselves. So in this case, we're doing an extreme zoom. You can see that the left edge of this part D of this figure ranges from only 917.98 up to 918.02. It's a tiny little minuscule part of the mass to charge range. So we're looking very closely zoomed into one tiny little peak. And you can see that in each of these time points, each of these horizontal stripes across this figure represents a different time at which an MS scan was collected. And here the color, dark green being the most intense, and these lighter yellows being the less intense, these are telling us about how much intensity is observed for this peak in the original profile mode MS scan, which is to say that we're not just portraying this peak as a stick, we're looking at all of the intensity around it as observed in the mass spec. Okay, what we see is that there's this jogging back and forth of the center line, which is to say that in successive MS scans, the mass to charge value reported at the center of this peak differs a little bit. It's going to dance a little left and a little right to lower and higher m/z values at each time we observe it. So each of these observations is independent. Some of them come from a very intense signal, and some of them come from a very weak signal. We know that when the peak is intense, we get a better, more accurate estimate of what its mass to charge value is. But we also have another way that we can evaluate these. Now look how wide this m/z range is. 844.2 up to 845.8. What we're looking at is an isotopic packet. We have the monoisotope, which is like all carbon 12s, all oxygen 16s, and so on. We have what we call the C13 or M+1 peak, which represents a molecule in which probably one of the carbons has become is a natural heavy isotope of carbon. So I think that if I remember the numbers correctly, something like 1% of all carbons in the world, on Earth anyway, are carbon 13s. So some fraction of the peptide molecules have an extra neutron, or an extra 2 neutrons, or an extra 3 neutrons. And the spacing at which we see them in the m/z domain tells us what their charge is. So we have, in effect, multiple time points at which we are measuring this peptide. We also have multiple isotopes at which we are measuring this peptide. Both of those can be brought together to give us very good peak shapes to quantify how much of this peptide is present. However, MaxQuant goes one further. MaxQuant is able to quantify a peptide even if it hasn't been identified in the experiment. That might seem counterintuitive. How could you quantify a peptide if it has not been identified? The answer is, in all the other experiments, you knew the order in which identified peptides appeared. You've got the identifications. You can simply say, well, a peptide with this sequence appears here, a peptide of that sequence appears there, and you can track this across all of the experiments you're analyzing. And then, when you want to ask, gosh, I saw this peptide show up in experiment A, I want the intensity for it in experiment B. How do I do that? Well, you ask how the retention times at which the peptides were observed in experiment A compared to those same peptides as observed in experiment B. We know the one we're interested in wasn't identified in B, but we know what its neighbors were in the experiment A, therefore we can look for its location in experiment B and still integrate from the mass spectrum, because quantifying in the mass spectrum does not require a tandem mass spectrum to have been identified to that peptide. So that's an introduction, a very quick introduction to spectral counting and to extracted ion chromatogram integration. Detecting differences between cohorts at the protein level is quite routinely handled by spectral counting. If you simply want to know which proteins changed and in which direction between two cohorts, you really don't have to do anything more complex than spectral counting. On the other hand, if you are trying to detect differences at the peptide level, or heaven help you at the post-translational modification level, you're going to need to use chromatographic area because your information is no longer able to be summed across multiple peptides. For this, you need to be able to integrate the areas of a particular peptide ion across these experiments. And keep in mind that when you talk to a biostatistician, and you will, you are going to need to figure out a model that is appropriate to the data you have. If the data you have are count values, you do not want to be using something like a t-test. A t-test really requires values that are not stair-step like counts are. You observe 4 or you observe 5 spectra for this protein. So something like t-test kind of falls apart in a circumstance like that. If you're dealing with chromatographic intensities, you're probably going to have data that look much more continuous and thus appropriate for a t-test. But keep in mind, you may have missing values. If MaxQuant were not able to integrate that particular peptide in that particular experiment, what intensity do you write down for it? This is a question that has puzzled much greater minds than my own. So with that, we have our introduction to spectral counting and chromatographic intensities. In just a moment, we will move on to the final segment of our proteomics unit, Proteome Informatics unit, where we discuss isotopic labeling and targeted quantitation. See you in just a moment. And we're back. Let us make some discussion now of more advanced techniques for proteomic quantitation. For this, we're going to talk about isotopic labeling and targeted quantitation. Some basic principles, though, before we start. So, in isotopic labeling, we need to stress that we are not talking about radioactive isotopes, right? There's basically no mass spectrometrist on Earth that really wants to put radioactive stuff through his or her instrument. So what we're talking about are stable, heavy isotopes. So things like a carbon-13 is quite stable. It's not going to emit anything at anybody. And yet it does differ in its mass from carbon-12 by adding 1 Dalton. Okay. So extra neutrons do not change the chemical behavior of a molecule, generally speaking. So whether you are carrying carbon-13s or carbon-12s, this enzyme is able to do whatever it's doing. Mass spectrometry, however, is able to tell the light isotope from the heavy isotope for that sample, because they have different mass-to-charge values, naturally. So if I have a labeled sample and an unlabeled sample, I can mix them together and run them in the same tandem mass spectrometry experiment in order to provide data that combine the aspects of both. So if you have an internal standard that you are using in labeled form, you can add that internal standard to lots of unlabeled other samples and use it as a point of reference for quantitation. However, incomplete labeling can be a really big problem. If you have only 90% of the proteins in a cell culture labeled, and and then try to mix that with something unlabeled in order to quantify them versus each other, you're going to run into a big problem, because now your labeled sample is contributing some extra to the unlabeled channel. So that's going to complicate things for us. Let's talk about 3 different ways that isotopic labeling takes place. First off, metabolic labeling. Now, in the old days, a lot of folks called this SILAC, but that term's kind of been moved over to something else. So we'll just call it metabolic labeling for now. If I am growing my, my E. coli in a dish, I can provide it medium that provides a needed a needed element in only its heavy form. As a result, after generation of these heavy E. coli growing in plates that only give carbon-13 rather than carbon-12, for example, I have replaced essentially every carbon in those E. coli with the C13 that I've been providing them. Okay, so we can typically leave that with carbon or with nitrogen, but generally speaking, this is something where you are growing your samples in a very controlled environment and can determine where they get their elements from. These days, we typically see SILAC used to describe an experiment in which someone has incorporated a labeled amino acid that changes just that particular amino acid to a different mass. So you could imagine in lysine, for example, you might replace both nitrogens with a heavy nitrogen, and now we've got a heavy version of lysine and the lighter unlabeled version of lysine. Okay, so that's SILAC. Frequently, though, you're not able to control where the samples are getting their nitrogen or getting their carbon, for example. So in something like that, particularly like in human experimentation, we instead make use of something called an isobaric tag. And an isobaric tag is something where you digest up your original sample to peptides, and then you stick on to the N-terminus of the peptides you've produced these isobaric tags, often sold under the trade names iTrack or TMT, depending on the manufacturer. In this case, we do not see a change in the MS signal for the peptide. It's completely the same. You know, these other 2 techniques, you're going to have a pair of peaks, one representing the heavy and one representing the light. But in isobaric tags, you see just one peak there, no matter which isobaric tag has been labeled onto the peptide. The reporter ion that we use to quantify how much of one or the other is present is actually found in the MS. So these isobaric tags have the same mass for the MS measurement, but they provide different reporter ion masses in the tandem mass spectrum. Okay, so that's a little intro to isotopic labeling. I've added some papers here if you'd like to learn more about individual examples there. So how is this used? In this case, we have a sample 1 and we have a sample 2, and we want to be able to compare sample 1 to sample 2. But generally speaking, people are going to want to do much more than compare A to B. They want to compare a whole bunch of A's and a whole bunch of B's and so on. So for this, I want you to think of this internal standard that appears in both cases as providing a heavy labeled reference to which all these samples can be compared. So not just a sample 1 and a sample 2, but samples 1 through 20. All of them will get combined with some amount of this internal standard to be analyzed. We mix them, we do our cell lysis together, we do our proteolysis together, we do our deep proteome together, and then we do our identification in a way that allows us to identify both the heavy and light versions of each of these peptides. And we can put together a ratio comparing the amount of the unlabeled sample to our labeled reference. Those ratios are then combinable in a way that allows us to compare how much of sample 1 we had compared to sample 2, compared to sample 3, compared to sample 4, all because they've been compared to internal standards. Okay. So that's a brief take on isotopic labeling for proteomics, but what about this targeted proteomics thing? First off, I want to note that right now in the field of proteomics, there's a huge amount of momentum behind a topic called data-independent acquisition. Sometimes you'll see the name SWOT used in combination with that. And these represent ways that we can attempt to quantify all the proteins all the time. I am talking about something much more limited in scope that has a very long heritage attached to it, and that is very much the way to go if the number of proteins you're interested in quantifying is, say, 50 or less. If you want to quantify 1,000 proteins, this is not going to get you there. But if you want to quantify 50, it's much more robust and reliable than a lot of the other techniques we could talk about. Targeted proteomics answer the question, they answer the challenge that if you want a quantitative result, you do a quantitative experiment. Not one of these shotgun things, but rather an experiment that's designed to give you quantitative information with clear ratios and clear error bars for a limited set of proteins. So generally speaking, the interesting stuff in samples are not the proteins that are in the top 10 by intensity. That's just not the case. Most of the proteins we find as potential biomarkers in serum or plasma will be proteins that account for much less than 1% of the total sample intensity. So generally speaking, we're interested in stuff that could get masked by the presence of very intense proteins. Shotgun methods have a pretty low amount of reproducibility for an individual peptide. We frequently see that from shot to shot, from LC-MS to LC-MS, an individual peptide may be identified in 50% of the runs, and that might be all we've got from a shotgun perspective. Targeting, however, tells the mass spec instrument which peptides are of interest. As a result, it's not spending all of its time collecting tandem mass spectra of just anything. Instead, it's focusing the instrument cycle time on just the peptides you find interesting. So to do this gives us much better sensitivity for them. And we're able to do this in a way that gives us an intensity profile not just for the intact ion, like what you would see in MaxQuant, but also an intensity profile for the fragments produced from them. That allows us to play all these sources of information off of each other to give us much better quantitative precision for these peptides. These days, you will find that many proteomics journals will simply turn your paper down if you say, I did shotgun on these 20 samples of type A and these 20 samples of type B. These are the proteins that are different. If that is the thrust of your paper, you're quite likely to get shot down if you try to send that paper to Molecular and Cellular Proteomics or Journal of Proteome Research. So these days, having an orthogonal experiment, such as a targeted proteomics experiment to go along with your shotgun, is much more likely to get you something that's publishable. I've spent a lot of time now talking about the use of orbitraps, for example, to produce these shotgun inventories of complex samples. Very typically for targeted quantitation, we're going to use a much, much less expensive instrument, and typically one that isn't as heavily used, so you don't have to wait as long to get your results back. So all instruments, all mass spectrometers have an ion source and a detector and a mass analyzer in between them. The ion source is still just nanoelectrospray or microelectrospray, depending on which instrument you're using. They all have a detector that's going to translate the arrival of a packet of ions with a signal that we're going to use to create our chromatograms. But in this case, we're going to use what's called a triple quadrupole. This triple quadrupole gives us the ability to isolate particular ions at a particular mass to charge value, to collide them with gas, and finally, to measure the set of fragments that come out. This produces data that looks something like these. This is the Skyline software, which is very, very widely used for analyzing targeted quantitation. Here we see a particular peptide that's been observed in a library. Here we are able to see the chromatograms. You see the characteristic rise and fall of not just the precursor, but the fragment ions in these chromatograms here. and we're able to get peak areas out that allow us to make comparisons between cohorts. We can also see did the retention times of ions for a particular peptide change from one peptide to a peptide? Did we see a strong correspondence or a weak correspondence between precursors and fragments? These chromatographic areas are the peak quantities that we want to measure. So we're looking for a place where all of the Y ions for a particular peptide co-eluted, that they all appeared at the same time, which is this little band right here. We can, if we have enough mass spectra collected across these, we're able to fit a shape to this rise and fall of the chromatogram and get a very accurate area for it. Calibration curves are a kind of a challenging space to think about, but they're a really important one for anyone involved in a quantitative experiment. In this case, we are gradually increasing the peptide concentration over several logs from left to right. So this is a vanishingly small amount of the sample injected, and this is a huge amount at right. We also have a measurement that's been made from it, so we can compute the area of the chromatogram for each of these different levels. What we see is that from about a third of the way across to the top, as we increase the amount of sample we're injecting, we also get an increase in the peak area. But what's this business down at the bottom? What we see is that even when we're injecting a vanishingly small amount of the sample, we basically fall into the noise area of the mass analyzer, and so we are measuring some intensity, and even though there's there's essentially zero sample for it to be detecting. This is a problem for the limit of detection. So we need to be able to say kind of what is the baseline amount of intensity we must be able to see to claim that any amount of this protein is present. That's the limit of detection. And the limit of quantitation tells us about the degree of variability in these values. You can see that these red whiskers down here at the bottom are quite long, which is to say that our intensity measurement of these individual peptides is changing quite a lot, especially when we have a very small amount of that signal to measure. The linear response range, this area right here, shows where an increase in the amount of sample corresponds to an increase in the amount of our measurement. So these calibration curves are very useful to us in knowing the extent to which the quantitative information coming back from these experiments is worthwhile. One of the phenomena of mass spectrometers that's worth discussing before we leave this topic behind is the phenomenon that we're very, very accurate in our ability to determine the mass or mass to charge value of an ion. Very accurate. The 10 part per million claims about those are completely legitimate. However, we do not have the same degree of variation in our intensity measurements. Mass specs are tuned to produce excellent mass to charge accuracy, and yet the intensity is almost always measured in a way that amplifies variation. We frequently make use of things like electron multipliers, which say that an ion coming in to hit the detector creates a spray of electrons that hits another set of electrodes that kick off another spray of electrons until you have enough amplification that you have a measurable signal. This creates quite a lot of variability in the intensity you might measure for multiple scans of the same thing. So when we look at how this plays out in the realm of things like targeted quantitation experiments, in effect, we see that we may have a coefficient of variation of around 10% or 12% for targeted proteomics experiments conducted on triple quadrupole instruments like the ones we've described here. Now, a coefficient of variation is may seem like a bit of an unusual term, but it's a question about the repeatability of a quantitative measure. Here we're asking, what is the standard deviation for these repeated measurements divided by the mean value for them? In the case of the 10% CV that I'm describing here, you might take a look at this 2009 paper by Terry Adana and friends, which asked about the amount that the quantitative variability was, how much of this was observed when we looked at the same sample distributed to many labs equipped with triple quadruple mass analyzers. So this is an empirical number that reflects all of the variation that occurs when a different person is operating a mass spec in a different laboratory. So this was a true test of reproducibility, not repeatability, in that we were changing which instrument was in play, which person was operating it, and so on. So when we try to put all the messages together, we end up with these takeaways, I think. Labeling allows us to mix different samples together in a single LCMS-MS experiment. And this multiplexing gives us the ability to directly compare the intensities in a way that label-free experiments don't really get to do. Targeted quantitation, on the other hand, is something that it really requires us to say in advance which proteins we intend to do a comparison for. If you're able to do that, if you're able to say only these proteins from this particular pathway are of particular interest to me, then you can design a targeted quantitation experiment that will give you much lower variability in measurement than what you would expect if you did quantitation from a standard shotgun experiment. So I would really encourage people to make greater use of targeted quantitation experiments to improve their research. It really does have a lot of credibility in measurement to offer above and beyond what a simple shotgun experiment can do. False quantitation and intensity variation will always be with us, but we can estimate their effects. I don't know that I defined false quantitation all that well for you, but I would just note that it is certainly possible when you have a very small amount of a peptide present to essentially integrate the noise, to integrate just the background, to try to put together some sort of quantitative measure rather than having a missing value there. When we talk about systems like MaxQuant, using this retention time matching and so on, it is certainly possible that the software will give you a measurement of how much of a peptide is present in a sample when zero of that peptide is present in that sample. In a case like that, where you're creating an intensity, creating a measured intensity from background noise, you could certainly think of that as a false quantitation event. Being able to characterize the extent of this is quite valuable in understanding just how much to trust the numbers that come back from targeted quantitation, isotopically labeled quantitation, or plain old integration from shotgun data can seem to be. We are getting better with the statistical models that we use for evaluating the data from quantitative experiments. Well, I hope that this has been a useful couple hours of your time as we've talked about identification and quantitation in proteomics. If you're interested in the subject of post-translational modification, I would note that the webpage for picking up the tab also has a full lecture on post-translational modification. I really wish we could have fit it into these 2 hours, but I'm afraid we couldn't. So thank you very much for your attention. If you'd like to email me, I think my email address is still visible from Stellenbosch University or from Pasteur Institute, where I'm currently working. And hopefully next year I'll be speaking to you from South Africa. Thank you.